There's a silent threat lurking in our global food supply, and it's found in the very seeds and grains we store for the future. It's mycotoxin contamination. This is a critical vulnerability that can cause absolutely devastating economic losses and, even more importantly, poses a profound threat to public health all over the world. So today, we're going to break down the science of how we can manage this invisible threat.
All right, so here's our game plan. We'll kick things off by defining what mycotoxins actually are. Then we'll identify the specific fungi that produce them and the exact conditions they need to thrive. After that, we'll dive into their impact and how we even detect them in the first place, before wrapping up with the strategies we have to fight back.
So, first thing is first, to really tackle this problem, we've got to understand the enemy. What exactly are mycotoxins, and why are they such a huge issue after the harvest is already safely in the silo? So, put simply, mycotoxins are toxic chemical byproducts made by certain types of mold. You can almost think of them as the chemical weapons of the fungus kingdom. The scary thing is, they aren't alive themselves, but they are incredibly stable compounds. That means they can survive food processing and end up being a direct risk to us when we eat them. And this quote right here, it just hammers home the massive scale of this problem. We're not just talking about a few moldy kernels of corn. No, we're talking about a vulnerability that can actually cripple agricultural economies and spark major public health crises, affecting both people and the livestock that we all depend on. Okay, so let's get a little more specific. Let's identify the key culprits. Who are the main fungal offenders, and what are the specific toxins they're unleashing on our food? Well, it turns out that the vast majority of the mycotoxins we're really worried about, all the ones you see listed here, they actually come from just three main groups of fungi. These are Aspergillus, Penicillium, and Fusarium. And each one of these groups produces its own unique set of toxins, each with different dangerous effects. All right, let's talk about the big one, aflatoxins. These are probably the most infamous of the bunch. They were first identified way back in the 1960s after a huge incident. And aflatoxin B1, that's the really nasty one. The International Agency for Research on Cancer classifies it as a Group 1 carcinogen. That means we know, without a doubt, that it causes cancer in humans. And it's a huge problem in staple crops like maize and peanuts. See, and this just shows you it's not a one-size-fits-all problem. Here we have a totally different fungus, Fusarium, and it's making a completely different toxin called Fumonisin. These toxins target different crops, especially maize, and they're linked to a whole other set of terrible health issues, from esophageal cancer in people to fatal diseases in horses and pigs. This chart is fantastic because it lays out the connections so clearly. It's like a who's who of molds and their toxins. So for example, if you look at that top line, you see Aspergillus parasiticus. That little fungus is basically a factory, just churning out multiple types of aflatoxins. Mapping these relationships is absolutely critical for diagnosis and control. Okay, so we've identified the criminals and their weapons. But here's the thing. These fungi aren't producing toxins 24/7. So what's the trigger? What conditions flip the switch and turn a harmless little spore into a full-blown toxin factory inside our grain silos? Well, it really all comes down to the immediate environment, that tiny microclimate right there in the storage bin surrounding the seeds. Everything. The temperature, the moisture, even the composition of the air in that small space dictates whether the fungi will grow, and more importantly, whether they'll start producing these dangerous toxins. The single most important factor? It's water. But it's not just about the total amount of water. It's what scientists call water activity. You can think of it as the available water that microbes can actually get to and use to grow. For most grains, if that water activity level climbs above 0.70, you've crossed a critical line into the danger zone for fungal growth. And the second key trigger, of course, is temperature. Just like us, these fungi have a comfort zone, a temperature range where they grow the fastest and produce the most toxins. And for many of the worst offenders, that sweet spot is between 25 and 30 degrees Celsius. That's about 77 to 86 degrees Fahrenheit. I mean, that's a pretty common temperature for a storage facility in many parts of the world, right? And this data right here really puts it all together. It shows how those two key ingredients, water and temperature, create the perfect storm for each fungus. Let's look at Aspergillus flavus again, our aflatoxin producer. You can see it really hits its stride when the water activity is above 0.82 and the temperature is hanging around 30 degrees Celsius. If you can control these two factors, you can control the threat. So let's say the worst happens. The conditions are perfect, and contamination occurs. What does that actually mean for us? What are the real-world consequences? And how in the world do we detect these toxins that we can't even see? Well, the fallout is just huge, and it hits on two major fronts. Economically, we're talking about an estimated 1 billion metric tons of food getting affected every year. Contaminated crops become worthless, which means huge losses for farmers and even trade restrictions for entire countries. Then you have the health impacts, which are just as scary. We're talking everything from acute poisoning to long-term chronic problems like cancer, a suppressed immune system, and serious organ damage. So how do we find them? Well, it takes some pretty serious science. In a lab, we use highly sensitive methods like chromatography, which can separate a sample into all its chemical parts and find those toxins with pinpoint accuracy. But you can't always bring the farm to the lab. So for more practical, on-the-spot testing, there are also rapid diagnostic tools, which work a bit like a home pregnancy test, to give you a quick yes or no answer right there in the field. This brings us to the most important part of our discussion. Now that we understand the problem, what can we actually do about it? What are the concrete steps we can take to counter this threat and keep our food safe? The first thing to know is there's no single magic bullet. The only effective strategy is an integrated, multi-step approach that covers the entire post-harvest chain. It starts with a timely harvest, followed immediately by proper drying to get that moisture level down, then physically sorting out any bad kernels, managing the storage environment with incredible care, and finally, if it's already too late, using detoxification methods. And this slide right here perfectly illustrates one of the biggest game changers in this whole field: storage technology. Think about the old ways: jute sacks, clay pots. They breathe, right? They're porous, so they let in moisture and oxygen, which is exactly what mold and insects love. But modern hermetic bags, they are totally different. They're airtight. Once you seal them, the living things inside use up all the oxygen and create a high carbon dioxide environment that suffocates pests and just stops mold in its tracks. The difference in seed viability is absolutely staggering. And what's really exciting is that the future of control is looking to nature for answers. Researchers are finding that essential oils from plants like clove, turmeric, and ginger are incredibly powerful natural fungicides. And even cooler is the field of biological control, basically using good guy microbes to actively fight off the bad guy molds. This is a much safer and more sustainable way to protect our food. So when you step back and look at the whole picture, what's the big takeaway? It's that mycotoxins are this incredibly complex, deeply rooted threat sitting right at the intersection of biology, our environment, and the global economy. Eradicating this silent danger isn't about one single invention. It requires a holistic, integrated approach that combines smart harvesting, advanced storage technology, and innovative biological controls to safeguard our food from the farm all the way to our fork.